And so we are very pleased to listen to you. So I let you.
So thank you very much. There is an echo, maybe? No, now it's okay. So there are a couple of technical problems, so please do interrupt me if something is not going well. This has been a very nice conference, and I'm grateful with the organizers. Well, and I will talk about several projects that have in common that had been inspired by a famous experiment, which is known as the Lenski experiment. So what I will do is first give a very quick introduction to population genetics, but a very probabilistic way of thinking population genetics. Then I will talk about the Lenski experiment, and at the end I will just talk about a couple of problems and questions that I like. And now, of course, I cannot move the page. Yeah, okay. So let's start with the first model in population genetics, which is the Wright-Fisher model, or as I like to call it, the Wright-Fisher random graph. The idea is simple. You have individuals, so each of the points here, it's an individual, and at each generation, so a generation is a column, and at each generation you have a fixed number of individuals, n. Now the rule is that each individual is connected to its mother, and the selection of the mother is made uniformly at random and independently. And you just connect each individual to its mother to get by infinite random graph that we call the Wright-Fisher random graph. Once you have this object, there are basically two reasonable ways to study it. One is the frequency process, in which you take your favorite generation and you put colors. So let's say that I color some individuals in blue, and then I impose the rule that each individual copy the type of its parent. So this propagates the color through the time, and then I can define a stochastic process that counts the frequency of blue individuals at each generation. Similarly, you can travel your graph, your random graph, backwards in time. And this you do by fixing a sample in some generation. And now you see that the time goes in the opposite direction. So I fix a sample generation 0 here, and I start counting the number of ancestors as I travel to the past. So at the beginning, I have 3 ancestors because my sample is of twice 3. Then I have 2 because these 2 guys up here were brothers, and here I have only 1, and after I have 1 ancestor of everyone, I will always have 1. So this is what we call the time to the most recent common ancestor. Well, as the 2 processes live in the same graph, there is a way to relate them that we like to call sampling duality. The idea is simple. You have some colors, some frequency of blue individuals at generation 0, that is X, and some sample at generation G+1. And you want all your individuals in the sample to be blue. So what is the probability that all these individuals are blue? Well, there are 2 ways to answer this question. One is to travel backwards in time and follow the ancestors until generation 1. And in order for all these individuals to be blue, you need that all the ancestors at generation 1 have to be also blue. So this leads to this formula: the probability that all the individuals of your sample are blue are the probability that all your ancestors are blue. But you can also think about this in the opposite direction. So what is the probability that all the individuals in your sample are blue? Well, you can propagate the color blue until generation G using the forward process or the frequency process, and then let each individual choose from the previous generation. And this leads to this as the probability that all the individuals in your sample are blue. For sure, the 2 things have to be the same, and then we conclude something that we like a lot, and it's called moment duality, which is a relation between the ancestral process and the forward process. So this idea that if you know your past, you can say things about the future in this particular graphical model is actually completely true, and it's formalized in this formula. And the beautiful thing about this is that every time that you can put two processes in a graph like this, you have the same way of thinking to relate them in some duality. Well, there are many modifications that one can do. The Greit-Fisher model is very nice, but we can criticize it a lot. One way to generalize the model is to go to something called Canning's random graphs, which are very similar to Greit-Fisher random graphs, except that individuals choose their parents uniformly at random from the previous generation, but this uniform doesn't have to be independent anymore. So it could be that there is a very fit individual and everyone chooses this fit individual, but the way you choose the fit individual is random, and then you are in the Canning's world. Well, let's now take a look of what happened in the Greit-Fisher world again when you take population size to infinity. So in mathematical population genetics, something that we like to do in order to deal with the complexity of a lot of individuals is to take limits. And well, it's well known that if you have n individuals at each population and you measure the time in units of the population size and send n to infinity, so the population size to infinity, you have a convergence to an object that is central in the theory, which in this case is the block counting process of the Kinman coalescent. This is a very simple process in which each pair of individuals coalesce at rate 1. So you lose an ancestral line at rate n choose 2. And something interesting here is that, well, even if you start with a huge sample, you will have a lot of coalescence, and after a very short time, you will find a common ancestor. And this idea of finding a common ancestor, it's dual to the idea of the frequency process being neutral. What do I mean with this? So if you go to the forward picture, you have that the frequency process in the same scale converges also to something very classic, which is the Greit-Fisher diffusion. What is the first thing that we like about the Greit-Fisher diffusion is that it's a martingale, which means in particular that if you start in 1/2, the probability of ending up in 1 is 1/2, and the probability of ending up in 0 is 1/2. So going back to the interpretation of this guy, it means that if you have two colors competing, the probability that one wins or the other wins only depends on the initial frequency. And this is what we call neutrality, right? So you go to up with the same probability that you go to down if you start in 1/2. But let's say one or two more things about this guy here, because it's a very nice piece of theory of probability, but in particular of population genetics. So a diffusion is the solution of a stochastic differential equation that has this form like here. I don't want to go to details, but the red part will be the noise, and the blue part will give you the deterministic part. And this will be very helpful to model all sorts of things, in particular in population genetics. So this looks something like this. The blue part gives you information that you need to do when you are, for example, if you are staying here, the blue part tells you to go down in a very similar way than an ODE, like a ordinary differential equation. And the blue part we can think as Gaussian noise in each of the points where you are sitting. So the way I represented this is, if you have a lot of red, it means that you have a lot of noise. So maybe you start here, and then the noise makes you move a bit. And then when you start in this region, the blue part takes you up. And then the noise maybe takes you down. And here, the blue part wants to take you down, and there is no noise that can stop this to happen, right? So red part, noise, blue part, deterministic movement. In the Greit-Fisher diffusion, what you have is that the blue part doesn't exist. So it wants you to stay in the same place, and you only have noise. and there is a lot of noise in the middle and very few noise in the corners. There is a very nice paper that explains all of this by our friend Peter, who gave a very nice COVID related talk. And if you want to learn more about this, well, take a look to this paper. Now, there are many ways to generalize the Greit-Fisher model. And in particular, something that we might not like is that so far it's neutral, as we discussed. So each of the colors had the same probability of winning, but usually we're interested in cases where there is selection. So where one type is better than the other somehow. And there are different ways to model selection, which are good in different situations. I will explain one way which generalizes the classic Greit-Fisher selection, and that I studied with Darius Panos some years ago. I like this because it's still in the graphical world, and the idea is quite simple. So each individual chooses its parent, but now it will not choose only one, but it will choose a random number of individuals from the previous generation. So for example, this one chooses 2, this one chooses 3, this one chooses only 1, and there is a random variable that tells you how many observations do you have in the past. And then selection is introduced by saying that you choose the best type that you find. So for example, in this case, let's say that blue is better than yellow. So this first individual will be yellow because it's only connected to yellow individuals. But this individual here will be blue because it's connected to a yellow individual and it's connected to a blue individual, and it chooses the blue individual as its parent. This way you propagate the colors, and it's very nice because you have a graphical representation. So if you think about it, you don't really need the colors in order to construct the graph of the ancestral parents, and then you can have a forward process and a backward process, and these guys will be dual. Also, the forward process in the same scale, under some hypothesis, converges to a diffusion that is the Greit-Fisher diffusion with selection. So if you look at this, it's exactly the same diffusion that you had before in this second part, but in the first part, now we have a drift. Now we have a blue part that it's helping the blue individuals, and it's asking the yellow individuals to go to extinction. So it looks something like this. Now the deterministic part is pointing downwards, and the noise part is exactly the same as before. Something that we like about diffusions is that they allow us to calculate probabilities of fixation. And you see here that the probability of fixation of the yellow individual, who was the not favorable one, is decreased a lot by this selective disadvantage, that here I put a huge selective disadvantage. And for example, in this case, in order to have half of the probabilities of fixation for the yellow individuals, you would need to start with 0.9 frequency of yellow individuals. Okay, the dual process looks something like this. So you start with a sample and you start traveling backwards in time, and you have now coalescence as before, but you also have, so here's a coalescence, you also have branchings due to the potential, to the many potential parts. And the two guys are dual, but also in the limit, the diffusion, the Greit-Fisher diffusion, will be dual to some also very famous process, which is the block counting process of the ancestral selection graph, which is again a very simple process where blocks coalesce at rate 1. So each pair of blocks coalesce at rate 1, and each block branches at some rate s. So it looks like this. It was introduced by Krohn and Neuhauser in '96, and it just looks like this. So you can have coalescence, and maybe you reach 1, and in 1 you can have a branching. So 1 is not an absorbing state as before. And this fact that is not absorbing state is the way that the dual look at selection. So if one is absorbing, like in the Kidman coalescent, then you have neutral. And if it's not absorbing, then you will have something different happening, in particular here, selection. Well, let's go to the second part of the introduction, where I will describe the Lenski experiment. So the Lenski experiment is an important experiment in experimental evolution, started by Richard Lenski in 30 years ago. And his idea was to have a very simple system in which he could measure and see evolution in action. So what he did was putting 12 independent repetitions of Escherichia coli in minimal glucose medium. And what he's doing is, do you see this correctly? It looks weird in my...
It is small. We can see it, but it's very small suddenly.
I don't know how to change this. Is better?
Yes. Yeah, but it's just a bit on the right. Yeah, perfect idea.
Now it's too big, right? Now it's okay?
Yeah, yes, it's okay.
Perfect. So the idea of the experiment is that they in the Lenski lab put small amount of Escherichia coli at the beginning of the day, like 5 million of cells. And during the day, the bacteria reproduces and eat the glucose in the media. And then, well, the glucose is finished and the bacteria just reach a maximum capacity, which is like 100 times the starting number of cells. And then N, so the population is taken back to 5 million by a uniform sampling. And well, a new day start by putting this sample in fresh media. And the very cool thing about this, well, there are many very cool things about this, but one is that this has been going on for 30 years. So the bacteria had really learned to play this game. And something also very important is that they have samples of the history of the evolution of this bacteria stored in fridges. So you can go and compare the bacteria from 10 years ago with the bacteria today, or the bacteria from today with the one at the beginning of the game. And this is very important for many reasons, but one of them is that it's allowed them to quantify the adaptation. So if you think about it, quantifying the adaptation is not easy. And one thing that you need is a fixed medium in order to say who is more adapted, because you need to be adapted to some particular conditions. And also you need a way to compare different strains. And all of this is possible in the Lenski experiment. So they come with the notion of relative fitness, which is simply put your strain from today in the same media as a strain from the very beginning. So you put a small amount of each in your fresh media. You wait for the whole day, and you see the proportion of each of the types at the end of the day. And this, well, you take the logarithms of that because there is exponential growth, but this gives you a measurement of adaptation that is called the relative fitness. And something that they observed is that, well, the relative fitness is increasing. So there is evolution somehow. There is adaptation to this game of daily cycles. So after 10,000 generations, you can see that the relative fitness have increased. And this happened in the 12 replications. After 20,000 generations, you see the green curve. Well, now there is even more adaptation, and it seems to keep increasing. And then at 50,000 generations, you still see adaptation going on. So the curve keep increasing. And what was very surprising for for people in the Lenski lab and co-authors was that it doesn't seem to be the case that there is a plateau. So they prove that the best way to approximate the curve of the relative fitness is by a power law curve that, in particular, if you put infinite here, what you have is infinite. So if you give infinite time so far, you would expect that there is infinite adaptation, right? So there are many biological reasons for this not to be possible, but what this means is that so far there is no sign of a plateau of adaptation coming. And now this looks huge. Let's see, sorry for this. Okay, so what is causing this deceleration in the relative fitness? People in the Lenski lab had basically two ingredients that used to explain this deceleration. One of them is epistasis, which is a concept that I don't really want to discuss a lot because it's too wide for me, so I don't quite understand it well. But the other is clonal interference, in which you have that different strains arrive and they want to go to fixation, but they do not compete with the basic population, but they start competing with each other, right? So maybe you have two beneficial mutations that don't compete with each other, but they need to overcome, they don't compete with one basic unevolved strain, but they need to compete with each other. And this for sure should cause a delay in the adaptation. Well, some other thing that I found very interesting about the experiment is what happened to the cell size. So this is not looking very well in my computer. I hope that it looks better in yours, but basically what they observed is that the size of the cells increased in all the 12 repetitions, right? And well, there should be maybe biological reasons for that, but I was a bit surprised that this was the case. So cells tend to grow bigger. Another thing that happened in all the 12 replicates is that the lag phase became short. So this means that when you put the bacteria in the fresh medium, the bacteria will not start reproducing immediately, but it will take some time. And then after this time, it starts growing exponentially. Well, what happened is that in the 12th repetition, the time it waits until start growing like crazy became shorter and shorter. Something also very puzzling for me is that in 7 out of the 12 replications, mutators appeared. So what does this mean? So if you see this part, the B part of the figure, you see the mutations on the 12 replications, and you see some lines that just go to the sky. This is when the mutators come, and they start mutating much more than usually. So you have this small square where you can see in the right scale the mutators, but this happened a lot. And what is very impressive is that the relative fitness that is up here in figure A doesn't seem to realize that there is a huge change in the biology of the cells. Well, this leads to some questions. The first one is about the consumption of citrate, because it also happened in 1 out of the 12 repetitions that the bacteria learn to eat something that is not supposed to be able to eat. So in the medium there was citrate. The bacteria, the Escherichia coli, doesn't eat citrate, but it decided that it was a good time to try this in 1 of the 12 replications, right? From my side, I am amazed by these key mutations that really change the picture and that live in the realm of the large deviations and not in the realm of the law of large numbers. But this is something I would like to understand. Also, I would like to understand the emergence of mutators and how this doesn't seem to change the adaptation, right? The rate of adaptation. I would like to understand the deceleration of the relative fitness. Why? I mean, Okay, it goes to infinity, but clearly it's deceleration. It's decelerating. What are the causes of this? Also, a good question is, how does the change in the lag phase can be modeled from the population genetics point of view? Because, well, this will give you a very different way of selection. I am also puzzled by the cell size. I mean, why does it increase? Is it a good idea to be bigger in a media where you are in a liquid media where you are just moving around randomly? And the last point is clonal interference, right? So how are we going to model clonal interference? Many of these points have been studied by different authors. And what I want to do for the rest of my talk is to, well, the first two I have never touched. So it's more an invitation if somebody wants to think about this. I think it's a lot of fun to read these Lenski related papers and just get some ideas. And the other four, well, are things that I have worked on directly or indirectly. And I want to talk about these four things. Okay, so the first problem I want to discuss is the expected curve for the relative fitness. So we see clearly that that there is a deceleration. We know from the Lenski lab that there are factors that cause part of this deceleration, which are epistasis and clonal interference. But my question was, or our question was, if we only have easy stuff, so if we only have the design of the experiment, what curve would we expect? So how much change is done by this epistasis and clonal interference? Okay, now again it's small. Sorry about this. Well, and in a project together with Noemi Kurt, Anton Bakolbinger, and Yin-Long Zhuang, what we did was to construct a very simple model, so the simplest possible model in which we capture, or we try to capture, the design of the experiment. The model works like this. So at each day, there are n individuals at the beginning of each day. Then individuals start splitting at a constant rate. And then when you hit 100 times n individuals, the reproduction stops and you sample n out of the 100n individuals. So I need to do this by hand. So the model looks like this. You start with some n individuals, then the individuals reproduce by pairwise branching, and they reach 100n individuals. Then you sample n out of the 100n, and then you do the same again and again and again. And this is a simple model that resembles just the experiment, just the Lenski experiment. So we were happy because one thing that you can do is connect the beginning of each day with each other and forget about what happened in the middle. And in this case, what you get is a Canning's model. And Canning's model are classic, and we can study them. And then we are happy because our model lives in the classic theory of population genetics. Because of this, also, we can add selection. So imagine that red individuals reproduce at R plus something small, and blue individuals reproduce at rate R. So what you will have is that there are typically bigger red trees than blue trees, and you sample just as before. And what you have is, well, the same model, but now you have selection because red individuals will tend to win over blue individuals. Okay, so something that we managed to do in this model is, if you have these two types, one reproduces at rate R and one is a little better, you want to know what is the selective advantage of this kind of reproducing faster mutation in our model. And this led to a formula that I like a lot, which says that if you start with one mutant, so one guy that reproduces slightly faster, you expect to have at generation one mutant plus this thing here, which is the difference on the reproduction rates, and this is thing here, that it's the time they needed to wait until resources were deployed. And why do I like this formula? Because if you see, there is an R down here, and R is the reproduction rate of the basis population. So this is already a story of deceleration, right? So if R is very small, like in the beginning of the experiment, then the selective advantage will be big. But if R is very big, so if the population is already adapted, then this selective advantage will be very small. Why is this the case that this is happening? Well, because the individuals don't have a whole day to compete. They only compete in our model until the glucose is over. And the glucose is over in a time that depends on how fast you eat the glucose. So if you eat it super fast, you will have very little time to compete, and then reproducing a bit faster is not such a great advantage. And now we go back to big. So what we did next is consider a model in which there are a lot of mutations. So mutations come each random time, and each of them gives you some small selective advantage. And we define some stochastic processes, which is the reproduction rate of the basic population and the relative fitness, the relative fitness at such time. For our model to work, we need to work under assumptions of no clonal interference, meaning that one mutation comes and then it either goes to fixation or dies before the next mutation comes. So it looks like this. A mutation comes, imagine that you are in position X, so it means that your relative fitness is X, and then a mutation comes and makes someone reproduce faster. This person will increase in numbers, but maybe with some probability it just dies, and then a new mutation comes and the same, a new mutation comes and the same, and at some point a new mutation comes and pop, it goes to fixation. It turns out that we can calculate the probability of fixation if we know the selective advantage of one mutation, and it's inversely proportional to this X, right? So the number of attempts that you will need to do before you see a success, before you see a fixation, will be inversely proportional to this X, and this again is the story of deceleration of the relative fitness. And these are the main ideas that lead to this theorem, in which we, in the right scale, that for sure depends on the mutation and the selection, so the mu is the rate at which new mutants come, and in this scale, we have convergence to a deterministic curve, so the relative fitness behaves like a deterministic curve, and this curve is nothing more and nothing less than a parabola, right? So if you look carefully to this, it's just 1 plus a constant times t, right? And this is already saying that there is a story of deceleration hidden in the design of the experiment, right? And all the other things, epistasis and clonal interference, well, they have to work on top of this deceleration caused by the design of the experiment. And now my slides have disappeared. I don't know what happened.
Yeah, because you put plus and not minus. Maybe with minus it will work a bit better.
You are a genius, Charlene. Thank you. Okay, so this can be found in this paper, and now in this other, what we did was trying to quantify how much of the deceleration comes from the design of the experiment and how much comes from the clonal interference and epistasis. And we have this graph. So this is before adding clonal interference or epistasis. What we see is that the red curve is the real data, and the blue lines are simulations with our model. and you see that the fit is actually rather bad, right? But if you add the extra ingredients, clonal interference and epistasis, then the fit becomes extremely well. Well, this is not a great result for me. I mean, it's nice, but it's an invitation to understand more what is happening in the experiment, because I like clonal interference, but epistasis there are many different ways of having epistasis. So I would like to understand more what other things are causing the deceleration in this Lenski experiment. And one thing that for sure has to play a role is the selection in the lag phase. So there are some mutations that are different from mutations that change the Malthusian, which are the mutation that change how fast you wake up. And these are very special mutations because they occur at the beginning of the experiment. And once you have made your lag phase close to 0, then these mutations will not be helpful anymore. So there is a strong selection on making the lag phase small, but this strong selection occurs only at the beginning of the experiment. And I conjecture that this can help to make a better fit to the real data. But inspired on this lag phase story, we work on a model in which we want to understand how the stochasticity of the lag phase change the genealogy of a given population. So imagine that you have some cells and you put them in fresh media, and they wait some exponential time before they wake up. And when they wake up, they start reproducing exponentially. And then at some point, you sample some of them. So at the beginning of next day, you always have the same amount of cells. So I want to know, how does the randomness on when do you wake up change the genealogy of these populations? And this leads to this theorem that is just in the archive and was written together with Maite Wilke-Berenger, Fernando Cordero, and Jason Schweinberg. And it's saying that the thing that you need to look at is how fast do the individuals reproduce when they are awake, and the parameter of the exponential. And this gives you some letter A that helps you to distinguish different behaviors for the genealogy. So if A is big, you will have that the genealogy is just the classic Kinman coalescent. But if A is between 1 and 2, something very nice happened, which is that you have that the limit is a beta coalescent. And if A is before less than 1, then you have one of these discrete coalescent. This theorem has the same flavor as the theorem for the branching process of Jason Schweinberg in 2002, but it's quite nice, I think, because it gives yet another way in which you can actually find lambda coalescence in the reality, right? For sure, the more important one is that they occur in the context of selection, and this was observed by Jason Schweinberg a few years ago. So the lambda coalescent, it's an object that had been studied a lot mathematically, introduced by Pittman and Sagitov, and is similar to the Kinman coalescent, only that now many blocks can merge in these big merging events. So this story that is also considered more things is in a preprint with Fernando, Jason, and Maite. And well, the work in progress is now we consider only the randomness on when do you wake up. But we would like to consider if this can be something that you learn from a mutation. So how those mutations that make you wake up earlier increase your relative fitness. And this for sure will take us back to look at the data of the experiment and see if our fit is good and if we can explain the deceleration only with the experiment, clonal interference, and the lag phase, right? So this is the dream so far, but we're working on this. An open problem that I think would be interesting I mean, I definitely need help for this, and maybe somebody knows how to do it, is I want to know what is the effect of the design of the experiment on this deceleration. And there is a very easy, theoretically, way to think an experiment that doesn't have this deceleration, which is simply sampled before the depletion of the media. So if you sample before the glucose is over, you will have that the time that the individual have to compete becomes independent of the relative fitness. And an experiment like this would really say what is the effect of the design of the experiment on the deceleration of the relative fitness. Well, I am starting to run out of time, so I will be fast here, but another thing that I wanted to tell is about efficiency. So when I look at this picture of the cells getting fat, my question was, is it a good idea to eat a lot, or is it a good idea to eat less? So a population that is efficient with the resources will free a lot of resources that can be used by this population, but also by the competitors. And an inefficient population that needs a lot of resources will take resources away from it, but also away from its competitors. So is it a good idea to be efficient, or is it a good idea to be inefficient? This we studied using a modification of the Wright-Fisher model, in which now you don't have a fixed number of individuals, but you have a fixed number of resources. And each type of individual has a cost, and this cost can be 1 if you are inefficient, or can be 1 minus k if you are efficient. So in order to construct the next generation, if you know one generation, each individual chooses its parent uniformly at random, but then if it's purple, it's paying 1 unit of resources. And then the next it's also purple, so it pays 1 unit of resources. This one chooses to be white, so pays 1 minus k, and you continue so on and so forth until the resources are deployed. So this leads to a model, which is a Markovian model that can be taken forward in time as much as you want, and that converge in the same scale as always to a diffusion, which is, well, we also put selection on top of this, and it converts to a diffusion, which is the Wright-Fisher diffusion with selection. But now there is this pink part here that is telling the story of efficiency, right? And this pink part here turns out to be super interesting if you like duals, because it turns out that it does have a dual, and the dual is just the ancestral selection graph plus some pairwise branching term, right? So the dual is something that coalesce branches, and also 2 individuals can cooperate in order to have a branch, right? So it's very similar to the ancestral selection graph, only that now people can get together and branch. And what is very interesting for me in the biological sense is what are the probabilities of fixation of individuals that are inefficient. And what we see is that being inefficient is actually a neutral trait. So if you see the diagonal is the case when you have no selection at all. And if you have no selection at all, being efficient or inefficient is just neutral. But what is very impressive is that if you have selection, so here you have different lines, and the darker the line is, the more selection you have. And here you have 0 efficiency, so the cost of both types of individuals is the same. And what we see is, as we increase the cost, the selection is, or the probability of fixation increase radically in the presence of selection. So here is basically 1 always. So the moral of this is that efficiency plus selection makes you much stronger, right? Sorry, inefficiency plus selection. So there is a second order drift towards inefficiency hidden in this, well, in natural selection somehow, right? And this was very impressive for me because I don't know, I was thinking that efficiency was something good, but at least in these models, inefficiency seems to be the good evolutionary strategy. This can be seen from the dual, because if you don't have selection, then the process will eventually hit 1 and will be absorbed at 1. And this is the mark of a neutral story. But if you have selection, then you will always have this pairwise branching that will make you go upper and upper. And it's like if you have more selection. So this story is in 2 papers, one with Verónica Miropino and Juan Carlos Pardo, and the other with Juan Carlos Pardo and José Luis Pérez. And the open question here is there a graphical representation? So everything I showed before has a graphical representation, but this one, I have no clue how to come up with one. I mean, these pairwise branchings are destroying everything I believe on. I don't know if I still have 5 minutes, Charlene, or maybe I can stop here.
3 minutes.
Perfect. So the last thing that I want to show you super quickly is current work about clonal interference. So this picture on top is the clonal interference story. You see that everybody is fighting with everybody like crazy, and there is a lot of one strain being defeated by the other, and then a third one comes and defeats everyone. So our model that says there is no clonal interference is probably not extremely good, but the model of the biologist in the Lenski lab was, okay, this crazy interaction between strains go to a constant C inside of this equation W, right? So all this weird interaction becomes just a constant C. And I was very puzzled by this statement, but as many times is true, biologists are right. You just need to understand how to prove their statement, and this is what we are doing now in our next Lenski project. So this is how I imagine clonal interference. You have a basic population, and then a new guy comes and start growing exponentially, and then a new guy comes and start growing, but at this point here, it needs to fight with the yellow and not with the red, and it's not better than the yellow, so it starts decreasing. And here this guy comes and it's actually better than the yellow and the red, and it will go to fixation. So the easiest thing that you can have is that a mutation comes and it's slightly better than the basic population, and you have exponential growth, and this is just a line in the logarithmic scale. But if you have a complicated picture, what you will have is many lines, and lines will refract each other. So when this line goes to fixation, the purple line is refracted, but it's still having a positive slope, but the yellow line is refracted, and it's now going back to 0. So how do we make mathematical sense of this? What we do is represent the lines and their slopes as points in R2. So each point is a time and a slope. And then we need to come up with an idea in order to work mathematically with the refraction of the lines. And this we do first by introducing an order, which is saying, if you have all these points here, which one will hit fixation first? And if you see this function is just time plus 1 over the slope. So it's just the time for its start, and then 1 over the slope is how much time do you take to go up. So this is sending each of the point to when does it goes to fixation. This gives us a pre-order in this R2, in these points of R2. And what we do is use this pre-order in order to choose the first point. And the first point is going to kick all the other points to model the refraction. And this kicking is simply take this point, which is this purple line, and sending to the prolongation of this purple line after it's refracted. So you follow the refracted line and continue all the way until it hits 0, and that's exactly this point. And this yellow point is just goes to 0. And this is a very nice way of ordering our set of points that leads to a deterministic dynamical system with values in the points. With this, we can have a notion of relative fitness, which is simply counting who's winning at each of the times up here. And this led to a theorem together with Felix Hermann, Andras Tobias, Renato Suarez dos Santos, and Anton Bakolbinger, which says that the relative fitness has a speed, which is a number between 0 and infinity. And at this point, the proof is only applying renewal theorem. And finally, thank you very much.
Thank you very much, Adrian. So does anybody have a question for Adrian? Okay, I have some, so I take the time. Yeah, I have a question. I remember that you told me that you had some exchanges with Lenski about the experiments. So finally, is it doing some? Is he doing some new experiments? And do you have still exchanges with him or no?
No, I mean, I have some communication with Phil Garish, who is a part of the Lenski group, and it had been a very nice exchange of ideas. But directly with Lenski, no, I don't think he's thinking deeply about these results. Maybe at some future time I can talk to him again.
Yeah, I had a question also about yeah, you were very brief about the epistasy and clonal interference. So one of the paper in theoretical population biology or another theoretical biology, I don't remember. How did you model epistasis and clonal interference in this work?
Yeah. So clonal interference we actually model by simulation. So if you look at this picture, maybe I can go back to it. If you look at this small picture, that maybe I will find it, maybe not. Right here. So there are a lot of green trajectories that actually are different simulations where clonal interference occurs, right? So simulating clonal interference is not that hard because you just let one mutant to start spreading and you allow a second one to come and they start fighting naturally, right? And epistasis, this is why I don't like epistasis, because it's something that you impose to the model a priori. So you start giving mutations that are smaller as the fitness goes up, right? So imagine that at the beginning of the game, I give you 1 peso, and if you already have 100 pesos, I will give you only 0.5 pesos, right? So then obviously there is a deceleration because your model has the deceleration a priori.
So yeah, you put in the model what you want to observe at the end in some sense.
Yeah, this is basically why epistasis is my enemy. I mean, because it's not, I mean, I understand that there are biological reasons for this, but from the modeling perspective, it's not telling you anything. It's not explaining you what is happening.
Right? Thank you, Adrian, for your presentation. Can you tell us a bit more about the growth on citrate that you mentioned at the beginning?
About so the what?
The growth on the use of citrate?
Well, I don't know. I don't know a lot. I mean, I can tell you what I know, but what I know is that is a great thing to look at.
Yes, I agree.
There are a lot of videos by Richard Lenski and his collaborators. What happened is that citrate is used to make the medium resist, so it's conservative.